Biomimicry, using nature to grow materials. Civilization is based on the human drive to invent. We take the raw stuff of our planet, the materials that give names to the ages, and craft them into new forms. However, this comes at a cost. Technological advancement has led to the large-scale production of materials and in the process has adversely affected our ecosystem. The manufacturing process for synthetic materials can require high temperatures, low pHs or highly acidic conditions, toxic chemicals such as heavy metals, and can use extraordinary amounts of energy. For all our advancements in energy and materials production, we don't come close to the efficiency seen in nature. Nature has been perfecting her chemistry for 3.5 billion years, and natural selection plays for keeps. Species pay the ultimate price for inefficiency. Biomimicry is the imitation of the models, systems, and elements of nature for the purpose of solving complex human problems. Living organisms have evolved well-adapted structures and materials over geological time through natural selection. Biomimetics has given rise to new technologies inspired by biological solutions at macro and nanoscales. By modeling or adopting the mechanisms found in nature, companies are able to secure a strategic advantage amongst their competitors and possibly increase their margins. I want to take some time to talk about how nature makes materials, and finish by talking about how we can use these processes to make materials that we want to use, essentially hijacking nature to improve products like batteries, solar cells, and the generation of advanced fuel systems. One canonical example of nature's supremacy when it comes to making materials is the abalone shell. It is a biocomposite material that is 98% calcium carbonate. When we make calcium carbonate, we sell it as chalk. When you find mineralized calcium carbonate, geologists call it limestone. But nature uses it and a small amount of well-placed proteins to make a material that is 3,000 times stronger than its geological counterpart. When we think of building things, we use blueprints as plans, a foreman to interpret, and wood or steel for raw materials. Analogously, nature uses DNA for the plans, ribosomes are the foreman, and amino acids are the building materials to make proteins. Proteins carry out myriad processes to sustain life of any form. For the abalone shell, the proteins produced have a negative charge and are able to attract positively charged calcium. Calcium is a common element found in our ocean, and rigid shells provide a defensive advantage for protection from predation. But how can we use similar processes to make something with elements that nature hasn't had the impetus to assemble for some evolutionary advantage? MIT biomaterials researcher Angela Belcher is attempting to answer that question. She and her colleagues are using a technology called phage display to mimic the evolutionary process to select for proteins, allowing us to essentially use nature to grow materials. One promising application is to use this technology to grow nanowires that can be used as the cathode and anode for high-powered batteries. Some of the advantages here are that you are producing electrodes which are lighter and because they are at the nanoscale have more surface area per unit of volume than traditional batteries. A lighter and more efficient battery in a car, for example, can increase the car's range. It can also be produced at lower temperatures and operate at physiological pH, to name just a few advantages. Any battery requires three pieces: an anode, a cathode, and an electrolyte solution for ions to migrate between the two. The anode and the cathode are made of materials that exchange electrons through a circuit. Engineering batteries using nature follows the same principle. The difficult part is explaining it to people without an advanced degree in biology, but here goes. The process uses M13 bacteriophage, a benign virus that only attacks bacteria, to produce the desired proteins and self-assemble to form nanowires. A virus consists of single-stranded DNA on the inside and is wrapped in proteins on the outside. As you can see, the exterior proteins come in 5 different flavors. This makes for an attractive vector when trying to express different proteins. Here we see a diagram of the phage display cycle. Genetic engineering allows us to make the M13 virus with different proteins on their outer coats. A cDNA library is used to create many different versions of the bacteriophage, each expressing a different protein or a different peptide sequence on the outside of the virus. Following the production of the phage library, the viruses are mixed with the desired metal for the electrode. The M13 containing sequence which attracts the metal remain, while the rest are washed away in a process called biopanning. Think panning for gold, where only the desired metal remains. The process is repeated for multiple generations, each time selecting for the peptide sequence with the highest specificity for the desired compound. This rapid selection is akin to evolution, but it doesn't take millions of years. The engineered virus uses bacteria cells to create millions of copies of itself, and those copies are used to make the electrodes of the batteries. As you can see here, we have a prototypical M13 bacteriophage docking onto a classical bacteria, injecting the single-stranded DNA, where it is copied to produce double-stranded DNA, and is therefore able to replicate and produce additional copies of the bacteriophage containing the protein coat that is desired. Earlier generations, the P8 proteins attract iron phosphate, while the P3 proteins attract carbon nanotubes. The iron phosphate templated virus, along with the carbon nanotubes, aggregate to form the cathode of a high-powered lithium-ion battery. You can really think of this as very similar to getting burrs stuck to your coat when you walk through the forest. The P8 proteins are specifically designed and were selected as the ones that attract the iron phosphate compounds, ideal compounds for the production of cathodes, once again in these high-powered lithium-ion batteries. The researchers also used this method to create M13 viruses whose P8 proteins were attracted to a cobalt oxide material to make an anode. Currently, the group is attempting to engineer lithium-ion batteries using manganese oxide biotemplating on the M13 virus. This technology has shown promise in other applications as well, such as improving solar cell efficiency and photocatalytic splitting of water, which emulates photosynthesis and can be used in the production of hydrogen for hydrogen fuel cells. This is just one small example of the power of nature and how science is leveraging our understanding of genetic engineering, protein chemistry, and bio-nanotechnology to solve the complex problems we've created for ourselves.